Welcome to Circuit Buddy. Today we will be discussing about lac operon. Lac operon is a very important topic for class 12 biology. It's also important for Neet. So today we will be discussing about the various conditions in which lac operon gets activated and turned off. So first of all, let us understand the term operon. Okay. So what is operon? Now operon is a functional unit of DNA having a cluster of genes under a single promoter. So again I repeat it, operon is a functioning unit of DNA having a cluster of genes under a single promoter. Now why is it called lac operon? It is called lac operon because this operon is dependent on lactose for its activation, or in other words, we can also say the presence of lactose itself induces the production of the required enzymes which are essential for lactose metabolism. Okay, and that's why we can call it inducible operon. Okay. Now this operon was first observed by Jacob and Monod in 1961, and they were awarded the Nobel Prize for their contribution in 1965. Okay. Now let us understand the basics of operon. So the first gene is the regulator gene, that is the I gene. So it is called the regulator gene, regulator or inhibitor gene. Okay. Then we have the promoter region P, which can be divided into two parts. One part is for RNA polymerase binding, so it is RNA polymerase binding site, and another is for CAM complex binding site. Okay, so this is the site where CAM complex binds, and it induces a bend in the DNA, helping the RNA polymers to transcribe the structural genes. Now comes the operator. After that we have the structural genes lac Z, lac Y and lac A. The lac Z produces an enzyme which is called beta galactosidase. Beta galactosidase breaks lactose into its constituent monomeric units, which are glucose and galactose. You know very well that lactose, it is made up of glucose and galactose. Okay. Now the Y gene codes for an enzyme which is called permease, which helps lactose to enter into the cell, and after that we have this A gene which produces an enzyme which is called transacetylase. Okay. Now let us consider a scenario where glucose is present but lactose is absent. In this case, the regulator gene produces a protein which is called repressor. This repressor protein binds to the operator site, preventing the RNA polymers from transcribing these structural genes, as a result of which the lac operon is switched off. So it is in the switched off mode. Okay. Now consider a scenario where glucose is present along with lactose. In this case also, the cell will use glucose as a primary energy source. Okay, so in that case also this thing will happen. The lac operon will be inactivated or will be in the switched off mode. Now let us consider a situation where glucose is absent and lactose is present. In this case, the cell will utilize lactose as its source of energy. Okay, so in this case what will happen? The lactose will be converted to allolactose by the action of this enzyme, which is called beta galactosidase. Okay, so now this allolactose binds to the repression and induces a conformational change in the repression. Okay, as a result of which the repressor cannot bind to the operator region and the RNA polymerase can bind to the RNA polymerase binding site, and it can transcribe the structural genes, as a result of which we can get the products of these genes, which are beta galactosidase, permease, transacetylase. In this situation, the lac operon is in the switched on mode. So it is in the switched on mode. So the presence of lactose induces the activation of the operon, and as a result of which we can call it inducible operon. Okay, so this is all about lactose operon. Hope you found this video very helpful. Please like, comment and subscribe the channel so that you can get future notifications.